Hello students, today we are going to see 10th standard science chapter 20 breeding and biotechnology. இந்த லெசனோட புக் பேக் ஆன்சர்ஸ் பார்க்கலாம். அதுக்கு முன்னாடி சப்ஸ்கிரைப் பண்ணாதவங்க சப்ஸ்கிரைப் பண்ணிருங்க. First Roman letter, choose the correct answer. First one, which method of crop improvement can be practiced by a farmer if he is inexperienced? Option B answer, mass selection. Second one, Pushakomal is a disease resistant variety of dash. Option C answer, cowpea. Third one, Hemigri developed by hybridization and selection for disease resistance against rust pathogens is a variety of dash. Option D answer, wheat. Fourth one, the miracle rice which saved millions of lives and celebrated its 50th birthday is dash. Option A IR8. Fifth one, which of the following is used to produce products useful to humans by biotechnology techniques? Option D answer, both A and B. Enzyme from organism, then option B live organism. Sixth one, we can cut the DNA with the help of option B answer, restriction endonuclease. Seventh one, DNA is a option C answer, recombinant of vector DNA and desired DNA. Option C answer. Eighth one, DNA fingerprinting is based on the principle of identifying dash sequence of DNA. Option D answer, repetitive. Ninth one, organisms with modified endogenous gene of a foreign gene are also known as option D answer, both A and B. 10th one, in a hexaploid wheat 2n is equal to 6 into is equal to 42. The haploid n and the basic x number of chromosomes respectively are option D answer, n is equal to 21 and x is equal to 7. Second Roman letter, fill in the blanks. First one, economically important crop plants with superior quality are raised by dash, plant breeding. Second one, a protein rich wheat variety is dash, Atlas 66. Third one, dash is the chemical used for doubling the chromosomes, colchichain. Fourth one, the scientific process which produces crop plants enriched with desirable nutrients is called dash, fortification. Fifth one, rice normally grows well in alluvial soil, but dash is a rice variety produced by mutation breeding that grows well in saline soil. Answer, Atomita 2. Sixth one, dash technique made it possible to genetically engineer living organism, gene therapy. Seventh one, restrictions endonucleases cut the DNA molecule at specific position known as restriction site. Eighth one, similar DNA fingerprinting is obtained for dash, identical twins. Ninth one, dash cells are undifferentiated mass of cells. Answer, callus. 10th one, in gene cloning, the DNA of interest is integrated in a dash. Answer, vector. Third Roman letter, state whether true or false. If false, write the correct statement. First one, Raphano brassica is a man-made pteroploid produced by colchicine treatment. The statement is true. Second one, the process of producing an organism with more than 2 sets of chromosome is called mutation. The statement is false. The correct statement, the process of producing an organism with more than 2 sets of chromosomic is called polyploidity. Third one, a group of plants produced from a single plant through vegetative or a sexual reproduction are called a pure line. The statement is false. The correct statement, a group of plants produced from a single plant through vegetative or a sexual reproduction are called a clone. Fourth one, iron fortified rice variety determines the protein quality of the cultivated plant. The statement is false. The correct one, amino acid rich fortified rice variety containing more amino acids determines the protein quality of the cultivated plant. Fifth one, golden rice is a hybrid. The statement is false. Golden rice is a genetically modified plant. This is the correct statement. Sixth one, Bt gene from bacteria can kill insects. The statement is false. The correct statement, Bt gene from bacteria produces a toxin that can kill insects. Seventh one, in vitro fertilization means the fertilization done inside the body. The statement is false. The correct one, in vitro fertilization means the fertilization taking place outside the body by artificial means. Eighth one, DNA fingerprinting technique was developed by Alec Jeffrey. The statement is true. Ninth one, molecular scissors refers to DNA ligases. The statement is false. The correct statement, molecular scissors refers to restriction endonucleases. Fourth Roman letter, match the following. Answer for match the following for column A and column B. First one, Swanalica. Answer, semi-dwarf wheat. Second one, IR8, semi-dwarf rice. Third one, Scaccharum. Answer, sugarcane. Fourth one, Moong number 1, Pasholus mungo. Fifth Roman letter, understand the assertion statement, justify the reason given and choose the correct choice. First one, assertion. Hybrid is superior than either of its parents. Reason, hybrid vigorous is lost upon in breeding. Answer, option A, assertion is correct and reason is wrong. Second one, assertion. Colchinchin reduces the chromosome number. Reason, it promotes the movement of sister chromatids to the opposite poles. Answer, option D, both assertion and reason is wrong. Third one, assertion or DNA is superior over hybridization techniques. Reason, desired genes are inserted without introducing the undesirable genes in target organisms. Answer, option C, both assertion and reason is correct. Sixth Roman letter, answer in a sentence. First one, give the name of wheat variety having higher dietary fiber and protein. Answer, Atlas 66 is a protein-rich wheat variety. Second one, semi-dwarf varieties were introduced in rice. This was made possible by the presence of dwarfing gene in rice. Name this dwarfing gene. Answer, the dwarfing gene was got from a dwarf variety of rice from China named DGOU gene, DGWG. Third one, define genetic engineering. Genetic engineering is the manipulation and transfer of genes from one organism into another to create a new DNA called a recombinant DNA or rDNA. Fourth one, name the types of stem cells. Embryonic stem cells and somatic stem cell. Fifth one, what are transgenic organisms? Answer, first point, genetic modification refers to the alteration or manipulation of genes in the organism using rDNA techniques in order to produce the desired characteristics. Then second point, the DNA fragment inserted is called transgene. Plant or animals expressing a modified endogenous gene or a foreign gene are also known as transgenic organisms. Sixth question, state the importance of biofertilizer. Answer, importance of biofertilizer. The term biofertilizer denotes all nutrients, inputs of biological origin for plant growth. Example, artificial inoculation of rice with cyanobacteria to increase soil fertility. Significances. First point, they are eco-friendly. Second point, they do not cause pollution like artificial fertilizers. Third point, help to safeguard natural resources. Fourth point, they are cheaper and economical. Seventh Roman letter, short answer questions. First one, discuss the method of breeding for disease resistance. Answer, crop variety resistance to diseases. Wheat, hemigry, leaf and strip rust hill burnt. Then cauliflower, pusha shubhara, then pusha snowball K1 black rot. Then cowpea, pushpa komal bacterial blight. Second question, name 3 improved characteristics of wheat that help India to achieve high productivity. Answer, variety of wheat, then their characteristics. First one, hemigry, disease resistant variety. Then characteristics, resistant to diseases like leaf and stripe rust, then hill burnt. Then second one, Atlas 66, protein rich wheat variety. Then third one, Sonalika, Kalyan Sona, high yielding semi, then dwarf wheat variety. The above mentioned characteristics seen in different varieties of wheat helped India to achieve high productivity. Third one, name 2 maize hybrids rich in amino acid lysine. Shakik, Ratna, Proteina are maize hybrids rich in amino acid lysine. Fourth one, distinguish between option A, somatic gene therapy and germline gene therapy. Somatic gene therapy, germline gene therapy. First point, it is the replacement of defective gene as a somatic cell. For this germline gene, it is the replacement of defective gene in germ cells, egg and sperm. Second point, it is only beneficial to the patient but not carried to the next generation. For this side, second point, the gene can be carried to the next generation, but till date only somatic gene therapy has been targeted. Then option B, undifferentiated cells and differentiated cells. Undifferentiated cells, then differentiated cells. First one, for both, they are of variable potency. For this differentiator, these cells can carry out specific functions only. Second one, they are specialized mass of cells and can divide to form more cells or give rise to differentiated cells. They are specialized cell types and cannot form any other types of cell. They are formed from undifferentiated cells. Third point, example, embryonic stem cells. For this side, third point, example, adult stem cells. Then fourth point, for both fourth point, source of embryonic stem cells are early embryo. For this side, fourth point, adult stem cells can be got from amniotic fluid, bone marrow, etc. Fifth question, state the applications of DNA fingerprinting techniques. First point, DNA fingerprinting technique is widely used in forensic application like crime investigation such as identifying the culprit. It also used it for paternity testing in case of disputes. Second point, it also help in the study of genetic diversity of population, evaluation and speciation. Sixth question, how are stem cells useful in regenerative process? First point, sometimes cells, tissues and organs in the body may be permanently damaged or lost due to genetic condition of diseases or injury. Second point, in such situations, stem cells are used for the treatment of diseases, which is called stem cell therapy. Then third point, in treating neurodegenerative disorders like Parkinson's disease and Alzheimer's disease, then neuronal stem cells can be used to replace the damaged or lost neurons. Seventh question, differentiate between outbreeding and inbreeding. இதை நீங்கள் டிஃப்ரென்ஷியேட் பண்ணி எழுதிக்கங்க. இதில் ஒரே இதில் 2 பாயிண்ட் வருது. ஃபஸ்ட் பாயிண்ட், Outbreeding and inbreeding. First point outbreeding ku inbreeding ku same இருக்கு. First point it is the breeding of unrelated animals. Next first point it refers to the mating of closely related animals with the same breed for about 4 to 6 generations. Second point, the hybrids are stronger and vigorous than their parents. In this side second point, it helps to accumulate a superior genus and eliminated undesirable genus. Eighth Roman letter, long answer questions. First one, what are the effects of hybrid vigors in animal? Answer, objectives of animal breeding. Heterosis or hybrid vigors. The superiority of the hybrid obtained by crossbreeding is called as heterosis or hybrid vigor. Effects of hybrid vigor in animal breeding. First point, increased production of milk by cattle. Second point, increased production of egg by poultry. Third point, high quality of meat is produced. Fourth point, increased growth rate in domesticated animals. Example, gross breed of fowls. White leghorn x polymouth rock. Hybrid foal yielding more eggs. Then cross breed of cows. Developed by mating the bulls of exotic breeds and cows of indigenous breeds. Brown Swiss X Shahiwal. Second question, describe mutation breeding with an example. Answer, mutation is defined as the sudden heritable change in the nucleotide sequence of DNA in an organism. It is a process by which genetic variations are created, which in turn brings about changes in the organism. The organism which undergoes mutation is called a mutant. Two types of mutagens. The factors which induce mutations are known as mutagens or mutagenic agents. First point, physical mutagens. Radiations like X-ray, A, B and Y rays, then UV rays, temperature, etc., which induce mutations are called physical mutagens. Second point, chemical mutagens. Chemical substances that induce mutagens are called chemical mutagens. Example, mustard gas and nitrous acid. The utilization of induced mutation in crop improvement is called mutation breeding. Achievements of mutation breeding. Some achievements of mutation breeding are, first point, Sharbati Sonara. Wheat produced from Sonara 64 by using gamma rays. Then second point, Atomita 2 rice with saline tolerance and pest resistance. Third point, ground nuts with thick cells. Third question, biofortification may help in removing hidden hunger. How? Answer, first point, hidden hunger is a chronic lack of vitamins and minerals that often has no visible warning signs. People who suffer from it may not even be aware of it. Second point, this can lead to severe consequences. Hidden hunger may lead to mental impairment, poor health and productivity or even death. Biofortification. Biofortification is the scientific process of developing crop plants enriched with high levels of desirable nutrients like vitamins, proteins and minerals. Some examples of crop varieties developed as a result of biofortification are given below. First point, Protina, Shaki and Ratna or lysine rich maize hybrids developed in India. Second point, Atlas 66, a protein rich wheat variety. Third point, iron rich fortified rice variety. Fourth point, vitamin A enriched carrots, pumpkin and spinach. Fourth question, with a neat label diagram, explain the technique involved in gene cloning. Answer, gene cloning. First point, a clone is a genetically exact copy of an organism. Second point, in gene cloning, a gene or a piece of DNA fragment is inserted into a bacterial cell where DNA will be multiplied, copied as the cell divides. Then basic steps involved in gene cloning. First point, isolation of desired DNA fragment by using restrictions enzymes. Second point, insertion of the DNA fragment into a suitable vector, plasmid to make or DNA. Third point, transfer of or DNA into bacterial host cell transformation. Fourth point, selection and multiplication of a recombinant host cell to get a clone. Fifth point, expression of cloned gene in host cell. Using this strategy, several enzymes, hormones, and vaccines can be produced. Fifth question, discuss the importance of biotechnology in the field of medicine. Answer, using genetic engineering techniques, medicinally important valuable proteins or polypeptides that form the potential pharmaceutical products for treatment of various diseases have been developed on a commercial scale. Pharmaceutical products developed by or DNA technique. First point, insulin used in the treatment of diabetes. Second point, human growth hormone used for treating children with growth deficiencies. Third point, blood clotting factor are developed to treat hemophilia. Fourth point, tissue plasminogen activator is used to dissolve blood clots and prevent heart attack. Fifth point, development of vaccines against various diseases like hepatitis B and rabies. Ninth Roman letter, higher order thinking skills. First question, a breeder wishes to incorporate desirable characters into the crop plant. Prepare a list of characters he will incorporate. First point, higher yield. Second point, resistance to diseases. Third point, resistance to insect pests. Fourth point, short duration crop. Fifth point, tolerance to environmental conditions. Second question, organic farming is better than green revolution. Give reason. Answer, green revolution is the process of increasing food production through high yielding crop varieties and modern agricultural techniques in underdevelopment and developing nations. This has increased production and helped India tide over food crisis, but this requires high yielding variety of crops got by artificial method like breeding, use of fertilizers and pesticides. Some disadvantages have been listed below. First point, this promotes monoculture of certain desired plant varieties. Second point, use of fertilizers. First, pesticides in excess alters the chemical composition of the soil. Third point, all farmers cannot afford the cost. Fourth point, environmentalists felt that because of green revolution, the soil was affected and certain traditional crops varieties have been neglected. Organic farming. First point, in contrast, organic farming avoids utilization of synthetic fertilizers, chemical pesticides and make use of green manures, compost and biological pest control which do not harm the environment. Second point, further growing different plant species will lead to efficient use of nutrients and cause fewer outbreaks of disease. Third point, organic farming technique like intercropping use of companion plants will also increase biodiversity. Thus, organic farming is stable, sustainable and beneficial for future generations. Third question, polyploid are the characteristics by gigantism justify your answer. First point, polyploids are organisms having more than two sets of chromosomes. Second point, therefore, depending on the degree of ploidity, the number of genus get multiplied. Therefore, size of the fruits, vegetables are oversized. Third point, these fruits and vegetables also have a market value and their vitamin and mineral content is also high. Fourth question, P is a gene required for the synthesis of vitamin A. It is integrated with the genome of Q to produce genetically modified plant or. First point, what is P, Q and R? Then second point, state the importance of R in India. Answer, first point, P gene requires for synthesis of vitamin A. Then Q, plant whose genome is being altered by integrating gen P, or genetically modified fortified plant got up by integrating gen P in the genome of plant 3. Then second point, importance of R in India. The transgenic plants are much stable with improved nutritional quality, resistant to disease and tolerant to various environmental conditions. Objective, improved nutritional quality in rice. Then gene inserted, beta catenin gene. In human, beta carotene is required for the synthesis of vitamin A. Then gene inserted, golden rice, genetically modified rice can produce beta carotene that can prevent vitamin A deficiency.